Welcome, and let the weight of the day begin to ease away. Before we drift together into the world of nature's most fascinating hunters, I'd invite you to take a moment, if it resonates with you, to subscribe to this channel and turn on notifications so you never miss these peaceful journeys into science and wonder. There is something deeply calming about settling in like this, preparing to explore the quiet mysteries that unfold when we least expect them. Close your eyes for just a breath, or let them soften their focus, and imagine with me a landscape unlike any you might encounter in your daily wanderings. Picture a wetland at the edge of twilight, where the air hangs thick and warm, saturated with moisture that seems to shimmer in the fading light. The ground beneath is soft, almost yielding, a sponge of peat and moss that has accumulated over centuries, layer upon patient layer. This is a bog, a place where water pools but does not rush, where nutrients are scarce, and the soil offers little to sustain the roots that reach down into it. The earth here is acidic, thin, depleted of the nitrogen and phosphorus that most plants require to thrive. And yet, life persists. More than persists, it has adapted in ways that seem to bend the very rules we associate with the plant kingdom. You've felt hunger too, that pull toward what sustains you, that instinctive movement toward nourishment when your body recognizes a need. Plants experience something similar, though their hunger manifests differently from ours. For most plants, roots spread through rich soil, extracting minerals dissolved in water, absorbing nitrogen compounds that fuel their growth. But in these bogs, in these sun-drenched yet nutrient-starved wetlands, the soil offers almost nothing. The very ground that supports these plants betrays them, providing structure but withholding sustenance. And so, over millions of years, natural selection favored those rare individuals whose leaves could do what roots could not: capture, break down, and absorb nutrients from an entirely different source. This is the story of carnivorous plants, rooted beings that have learned to hunt. The evolutionary path that led to plant carnivory is not a single road but rather many parallel journeys, each undertaken independently by different plant lineages facing similar challenges. Scientists have identified at least 12 separate instances where flowering plants evolved carnivorous adaptations across 5 different orders of the plant kingdom. Each time, the driving pressure was fundamentally the same: a desperate need to supplement meager soil nutrients in environments where sunlight was abundant, but nitrogen and phosphorus were vanishingly rare. These wetland habitats, bogs and fens and seepage swamps, offer intense light for photosynthesis, ample water, and precious little else. For a plant to succeed here, it must find alternative sources of the nutrients that other plants drink freely from the earth. Think of it as an ecological trade, off a bargain struck with natural selection. The cost of producing specialized carnivorous leaves is substantial. These modified structures are expensive to build and maintain, less efficient at photosynthesis than ordinary leaves, and require energy to operate their trapping mechanisms and digestive processes. A Venus flytrap spends resources building trigger hairs and snap trap lobes instead of broad, light-gathering leaf blades. A pitcher plant invests in elaborate tubular structures, waxy surfaces, and digestive fluids, rather than simple photosynthetic tissue. In nutrient-rich environments, these investments would be wasteful, leaving carnivorous plants unable to compete with conventional species that channel their resources entirely into growth and reproduction. But in nutrient-poor bogs where nitrogen is the limiting factor, the equation shifts dramatically. The marginal benefit of capturing even a single insect, rich in protein and minerals, exceeds the marginal cost of maintaining the traps. A Venus flytrap can survive for 3 weeks on one large insect, its tissues enriched with nitrogen absorbed from the prey's broken-down proteins. Pitcher plants that capture numerous flies and ants throughout the growing season produce more leaves, more traps, more flowers, their reproductive success soaring beyond what soil alone could ever provide. In these specific habitats, carnivory transforms from an expensive curiosity into a competitive advantage. The origins of this carnivorous ability lie buried in the deep evolutionary past. Research using molecular markers suggests that the capacity for carnivory in the ancestors of the Venus flytrap emerged approximately 85 million years ago, during the late Cretaceous period when dinosaurs still roamed the Earth. The specific snap trap mechanism that makes the Venus flytrap and its aquatic cousin, the waterwheel plant, so distinctive evolved roughly 48 million years ago, a more recent but still ancient innovation. What began as simple defensive responses to herbivore damage, plants producing enzymes to deter insects from eating their leaves, gradually transformed into a means of capturing and consuming those very insects. This transformation required co-opting existing genes rather than inventing entirely new ones. The digestive enzymes that carnivorous plants use to break down their prey are remarkably similar to the defensive enzymes that non-carnivorous plants use to protect themselves from disease and pests. Chitinase, which breaks down the tough chitin in insect exoskeletons, evolved from enzymes originally designed to combat fungal infections. Acid phosphatase, which liberates phosphorus from organic molecules, was repurposed from its role in general plant metabolism. Even the protein transporters that allow nutrients to move from the trap into the plant's tissues are modified versions of the same transporters that work in roots, silently shuttling nitrogen and potassium from soil into the plant's vascular system. Natural selection worked not by creating entirely new biological machinery, but by redirecting and refining tools that already existed within the plant genome. Gene duplication. Most duplicate genes serve no purpose and eventually disappear, but occasionally a spare copy acquires a useful mutation that allows it to function in a new context. Over time, these modified genes enabled leaves to sense touch, to move rapidly, to secrete digestive fluids, to absorb nutrients through their surfaces, transforming ordinary foliage into sophisticated trapping organs. Interestingly, convergent evolution has guided different carnivorous plant lineages to evolve similar solutions despite their independent origins. When scientists examined the genes active in carnivorous plants across multiple families, they discovered that natural selection repeatedly acted on the same sets of genes, modifying them in parallel ways. The pitcher plants of the genus Nepenthes, native to tropical Southeast Asia, and the pitcher plants of the genus Sarracenia, found in North American wetlands, belong to completely unrelated plant families, separated by more than 100 million years of evolutionary history. Yet both produce similar digestive enzymes. Both evolved tubular pitfall traps. Both attract prey with colors and scents and nectar, because the fundamental problem they faced was identical, and the genetic toolkit available to solve it was constrained by the same biological limitations. This phenomenon, called parallel evolution at the molecular level, is relatively rare in nature, making carnivorous plants a particularly fascinating case study for evolutionary biologists. The extreme selective pressures imposed by nutrient poor ecosystems left little room for alternative solutions. If you are a plant that cannot move, that must photosynthesize in one spot for your entire life, and that spot offers almost no soil nutrients, your options are limited. You can evolve reduced growth and minimal nutrient requirements, accepting a slow, stunted existence. Or you can evolve the ability to extract nutrients from alternative sources, and in a wetland teeming with insects drawn to water and sunlight. Most accessible alternative. The carnivorous syndrome, as botanists sometimes call it, encompasses several distinct requirements. First, a plant must attract prey. Second, it must capture prey, employing mechanisms that prevent escape once contact is made. Third, it must digest prey, breaking down complex proteins, chitin, fats, and nucleic acids into simpler molecules. Fourth, it must absorb the resulting nutrients, moving nitrogen, phosphorus, potassium, and trace minerals from the external trap surface into the internal tissues. And finally, all of this must happen efficiently enough that the net gain in nutrients exceeds the net cost of building and maintaining the carnivorous structures. Different carnivorous plants have evolved different strategies for meeting these requirements, leading to a remarkable diversity of trapping mechanisms. Some traps are active, moving in response to prey contact. Others are passive, relying on structural features to prevent escape without any movement whatsoever. Some use adhesive substances to glue insects in place. Others employ pitfall designs, where victims tumble into inescapable chambers. The specific mechanism reflects the particular evolutionary path each lineage followed, shaped by its ancestral traits and the ecological niches it came to occupy. Among all carnivorous plants, none is more iconic, more instantly recognizable than the Venus flytrap. Its scientific name, Dionea muscipula, evokes both the Greek goddess and the ancient trap mechanisms used for catching mice and flies. Native to a remarkably small geographic area, a roughly 75-mile radius around Wilmington, North Carolina, this species grows exclusively in the nutrient-poor wetlands of the coastal Carolinas, in flat, moist, acidic habitats that receive full sun and experience periodic forest. It is the only species in its genus, a botanical singleton that has captured human imagination for centuries, since its first detailed description in the 18th century. Picture a Venus flytrap in its natural habitat. a low-growing rosette of leaves spreading close to the boggy ground. Each leaf consists of two distinct parts: a flat green petiole that performs photosynthesis like an ordinary leaf, and the remarkable terminal portion, the trap itself, formed by two hinged lobes that in their open state curve gently outward, like a book bent backward at its spine. The inner surfaces of these lobes are typically tinged with red, colored by anthocyanin pigments that may help attract insects by mimicking flowers, or perhaps simply by standing out visually against the surrounding greenery. Around the edges of each lobe, long, stiff bristles, called marginal spines, interlock when the trap closes, forming a cage that prevents larger prey from escaping while still allowing tiny, nutritionally insignificant insects to slip through the gaps. The true magic lies in the trap's inner surface, where 3 to 5 extremely sensitive trigger hairs protrude like delicate antennae. These hairs are the plant's sensory organs, capable of detecting the faintest brush of an insect's leg or wing. When you look closely at a trigger hair, you see a slender filament rising about 1/4 inch from the leaf surface, hinged at its base so that it can bend when touched. This mechanical displacement of the trigger hair activates ion channels in the cells at the hair's base, generating an electrical signal called a receptor potential. If this receptor potential is strong enough, it induces a full action potential, an electrical impulse that propagates across the trap, much like nerve signals travel through animal tissues. Here is where the Venus flytrap demonstrates a level of sophistication that borders on the uncanny. The plant does not snap shut at the first touch of a trigger hair. Instead, it counts. A single stimulation generates 1 action potential, but the trap requires 2 action potentials within approximately 20 to 30 seconds before it will close. This counting mechanism serves as a safeguard against false alarms, raindrops falling on the trap, bits of debris blown by wind. Even the plant's own movements as it grows might occasionally bend a trigger hair. But these stimuli typically do not trigger the hair twice in quick succession. A living insect, however, crawling across the trap surface in search of the nectar that glistens enticingly around the trap's rim, will almost inevitably touch a trigger hair multiple times as it moves, satisfying the plant's requirement for confirmation. Once 2 touches occur within the temporal window, a cascade of events unfolds with breathtaking speed. The accumulated electrical charge triggers a series of rapid biochemical changes. Proton pumps embedded in cell membranes spring into action, driving water movement that causes the trap lobes to shift from their curved outward configuration to a curved inward one. This is not a gradual bending, but rather a sudden snap through instability, a phenomenon familiar to engineers who study bistable structures. Think of an umbrella caught in a strong wind. The moment when its dome suddenly inverts. The Venus flytrap's lobes exist in 2 stable states, open outward and closed inward. The transition between these states happens violently and almost instantaneously. The speed of trap closure is among the fastest movements in the entire plant kingdom. From the moment the second trigger hair is bent to the moment the lobes slam together takes approximately 100 to 300 milliseconds, less than half the time it takes you to blink your eyes. High-speed video recordings reveal the drama of this motion, the lobes accelerating rapidly as they flip through the unstable midpoint, the marginal spines on opposite sides approaching each other with such velocity that prey has virtually no time to react. For an insect standing inside the trap when closure initiates, escape is essentially impossible. The lobes close before most insects can even begin to move their legs or wings in a coordinated escape attempt. What happens next depends on whether the trap has actually captured something worthwhile. If the lobes close on empty air or on a piece of debris too small to continue stimulating the trigger hairs, the trap will reopen after about 12 to 24 hours, ready to try again. But if the trap has successfully captured a living insect, the prey's struggles against the inner trap surfaces stimulate the trigger hairs again and again, each touch generating additional action potentials. The plant literally counts these subsequent touches. If 5 or more additional stimulations occur, confirming beyond doubt that the trap contains viable prey, the plant initiates the next phase, tightening the seal and beginning digestion. The marginal spines, which initially interlock loosely like fingers interlaced, press together more firmly as the trap lobes curve inward with increasing pressure. The trap transforms from a cage that might allow a sufficiently small insect to squeeze through into a hermetically sealed chamber. Digestive glands on the trap's inner surface, which have been quiescent until now, begin secreting a cocktail of enzymes into the sealed space. These enzymes include proteases to break down proteins, chitinase to dismantle the insect's exoskeleton, nucleases to digest DNA, and acid phosphatase to liberate phosphorus from organic molecules. The fluid that accumulates inside the closed trap is acidic, typically around pH 5, creating an environment conducive to enzymatic activity. Over the next 5 to 12 days, depending on the size of the prey and environmental conditions, the insect is systematically broken down. Proteins are cleaved into amino acids. Chitin is reduced to simple sugars and nitrogen containing compounds. Fats are hydrolyzed into fatty acids and glycerol. Nucleic acids are fragmented into nucleotides and eventually into phosphate groups and nitrogenous bases. All of these smaller molecules, now dissolved in the digestive fluid, are absorbed through the trap's inner surface. Specialized cells on the inner lobe surfaces take up these nutrients, shuttling them through transporter proteins into the plant's vascular system, distributing nitrogen, phosphorus, potassium, and trace minerals throughout the plant's tissues. When digestion is complete, the trap reopens, revealing a dried husk of indigestible material, the empty exoskeleton, and any parts too tough for the enzymes to break down. This dried shell is often washed away by rain or blown away by wind, leaving the trap clean and ready for another capture. But each trap has a limited lifetime. A single Venus flytrap trap can typically close and reopen 3 to 5 times, perhaps up to 7 times, if the captures are small and the trap suffers no damage. Each closing cycle, especially those involving full digestion, takes a toll on the trap's tissues. Eventually, the energetic cost of resetting the trap exceeds the benefit, and the trap senesces, turning black and dying back to make way for new traps that the plant continuously produces throughout the growing season. The Venus flytrap's trap mechanism is not merely fast but astonishingly sensitive. Recent research has shown that even a single slow touch, if it deflects the trigger hair sufficiently and at the right velocity, can generate 2 action potentials in quick succession, triggering trap closure. This adaptation may allow the plant to capture slow, moving prey such as slugs or larvae that might brush against a trigger hair only once but bend it through a large angle. The trigger hairs essentially act as both touch sensors and velocity sensors, integrating both the degree of deflection and the speed at which it occurs. Ion channels in the cells at the hair's base remain open as long as the membrane is mechanically stretched, so a slow deflection that maintains the stretch for an extended period allows enough ions to flow to trigger multiple electrical signals, even from a single contact. The ecology of the Venus flytrap extends beyond its spectacular trapping mechanism. These plants grow in habitats where fire plays a crucial role in preventing the encroachment of woody vegetation that would shade them out. Periodic burns clear away competing plants and accumulated litter, resetting the bog to an early successional state where Venus flytraps and other carnivorous plants can thrive in full sunlight. Interestingly, the trigger hairs also function as heat sensors. During a forest fire, the extreme temperatures cause the traps to snap shut, which may protect the plant's more sensitive inner trap surfaces from heat damage, increasing the plant's resilience to these periodic disturbances. Despite their fame and widespread cultivation, Venus flytraps in the wild face significant conservation challenges. The human population along the coastal Carolinas is expanding rapidly, leading to habitat loss through development, road construction, and agricultural conversion. Poaching for the horticulture trade has decimated some populations, as collectors illegally harvest plants from the wild, rather than growing them from seed or tissue culture. Changes in fire regimes, often due to fire suppression in developed areas, allow competing vegetation to overgrow flytrap habitats. Fertilizer runoff from nearby agricultural or residential areas increases soil nutrient levels, which paradoxically harms carnivorous plants by enabling non-carnivorous species to outcompete them. The U.S. Fish and Wildlife Service has been reviewing the Venus flytrap for endangered species protection, recognizing the precarious state of wild populations even as cultivated plants remain common in garden centers. In cultivation, Venus flytraps are remarkably easy to grow if their specific requirements are met. They need full sun, consistently moist, but not waterlogged soil, and absolutely no fertilizers or mineral-rich water. The substrate must be nutrient, poor and acidic, typically a mixture of peat moss and sand, and the plants must be watered exclusively with distilled water, rainwater, or reverse osmosis water, as tap water contains dissolved minerals that will accumulate in the soil and kill the plants over time. Given these conditions, Venus flytraps are robust and relatively fast, growing for carnivorous plants, producing new traps continuously throughout their active growing season, and flowering in late spring with tall stalks topped by small white flowers. Crucially, the plants require a winter dormancy period. In their native range, Venus flytraps experience cold winters where temperatures regularly drop below freezing. The plants respond by dying back to ground level, surviving the cold months as dormant rhizomes beneath the soil surface. Without this cold period, the plants weaken over time and eventually die, unable to complete their natural life cycle. In cultivation outside of their native climate, growers must provide an artificial winter by storing the plants in refrigeration or placing them outdoors where they will experience cold temperatures for at least 2 to 3 months. It is a curious thing, this plant that moves so quickly that counts stimuli, that digests flesh. The Venus flytrap challenges our intuitions about the boundary between plant and animal, demonstrating that responsiveness, memory of a sort, and predatory behavior are not exclusive to creatures with nervous systems. In studying the Venus flytrap, scientists have gained insights into how electrical signaling works in plants, how mechanical stimuli can be converted into chemical and hydraulic responses, how organisms can evolve sophisticated decision-making processes using only the basic building blocks of cellular biology. The cellular mechanism underlying the Venus flytrap snap is a marvel of biophysics that scientists are still working to fully understand. The trap lobes themselves are composed of multiple layers of cells with different properties. The outer epidermal layer and the inner epidermal layer respond differently to the electrical signals that trigger closure. When action potentials propagate across the trap, they cause cells in the outer layer to rapidly acidify, pumping protons into their cell walls. This acidification loosens the bonds between cellulose fibers in the cell walls, allowing the cells to expand quickly by taking in water. Simultaneously, cells in the inner layer experience the opposite effect, shrinking slightly as they lose water. This differential growth, outer cells expanding while inner cells contract, creates the mechanical stress that drives the rapid curvature change. The trap's geometry is crucial to this process. In its open state, the lobes possess a slight outward curvature, like a shallow dome. This curvature stores mechanical energy, much like a compressed spring. The trap exists in a metastable state, poised on the edge of instability. The biochemical changes triggered by action potentials provide just enough force to push the system past a tipping point. And once that threshold is crossed, the stored elastic energy is released all at once, causing the explosive snap through motion. Engineers call this a bistable mechanism, a structure that has two stable configurations and transitions rapidly between them. The Venus flytrap has evolved a biological bistable mechanism millions of years before humans developed the mathematical framework to understand such systems. Research into the reopening process has revealed equally fascinating complexity. Unlike the rapid closure, reopening is a slow process that can take anywhere from half a day to several days, depending on whether the trap captured prey. If the trap closed on nothing or on debris that provided no ongoing stimulation, it reopens relatively quickly, usually within 12 to 24 hours, resetting to its open configuration through a gradual reversal of the cellular processes that caused closure. But if genuine prey is present and continues to stimulate the trigger hairs, the plant commits to a different path. The trap seals more tightly, begins secreting digestive enzymes, and does not reopen until digestion is complete and nutrients have been absorbed, a process that typically requires 5 to 12 days. The digestive phase is where the Venus flytrap truly reveals its animal-like sophistication. The enzymes secreted into the sealed trap are remarkably similar to those found in the digestive systems of animals. The proteases break peptide bonds in proteins, reducing complex protein structures to chains of amino acids and eventually to individual amino acids that the plant can absorb. The chitinase specifically targets chitin, the tough polymer that forms the insect's exoskeleton, breaking it down into N-acetylglucosamine units and simpler sugars. This enzyme is particularly important because chitin represents a major component of the prey's body mass and contains significant amounts of nitrogen. Nucleases within the digestive fluid attack the DNA and RNA in the prey's cells, snipping the long nucleic acid molecules into shorter fragments and eventually into individual nucleotides. Phosphatases then cleave these nucleotides, releasing phosphate groups that are essential nutrients for the plant. The digestive cocktail also includes enzymes that break down fats and other lipids, ensuring that virtually every usable molecule in the prey's body is reduced to a form that can be transported across cell membranes and absorbed into the plant's tissues. The process of absorption is itself a sophisticated operation. The inner surfaces of the trap lobes are covered with specialized glands, sometimes called sessile glands, because they sit flush with the leaf surface rather than protruding like stalks. These glands are the interface between the external environment of the trap's interior and the internal environment of the plant's vascular system. Each gland consists of multiple cells organized to maximize nutrient uptake. The outermost cells of these glands possess membranes studded with transporter proteins, molecular machines that specifically recognize and bind to amino acids, sugars, phosphate ions, and other nutrients dissolved in the digestive fluid. Once bound, these transporters shuttle the nutrients across the cell membrane through a process that often requires energy in the form of ATP, the cell's energy currency. This active transport allows the plant to accumulate nutrients even when their concentration inside the cells is already higher than their concentration in the digestive fluid, effectively pumping nutrients uphill against a concentration gradient. The nutrients absorbed by the gland cells then move through the plant's vascular system, traveling in the phloem, the living tissue that distributes sugars and other organic compounds throughout the plant. Much as our circulatory system distributes nutrients absorbed from our digestive tract, the nitrogen obtained from captured prey has a profound impact on the Venus flytrap's growth and reproductive success. In experimental studies, plants that were regularly fed insects grew larger, produced more leaves, flowered more prolifically, and set more seeds than plants that were not fed. The difference in growth rate between well-fed and unfed plants can be dramatic, with fed plants sometimes achieving twice the biomass of their unfed counterparts over a single growing season. This demonstrates that while Venus flytraps can survive on nutrients absorbed from the soil and from atmospheric deposition in rainwater, Carnivory provides a substantial boost that translates directly into evolutionary fitness. Interestingly, Venus flytraps must carefully balance their investment in carnivorous traps versus photosynthetic leaves. The traps, while capable of photosynthesis, are less efficient at capturing light energy than flat, horizontally oriented leaves would be. The plant produces both types of structures, with the flat petioles of each leaf performing most of the photosynthesis, while the terminal trap portion captures prey. As the growing season progresses and the plant's nutrient status improves through successful captures, some plants shift their growth strategy, producing leaves with larger petioles and smaller traps, effectively reinvesting some of the nitrogen gained from carnivory into more efficient photosynthetic structures. The reproductive biology of Venus flytraps adds another layer to their ecological story. In late spring, typically May in their native range, mature plants produce tall flowering stalks that rise 8 to 12 inches above the rosette of leaves. This elevation is important because it separates the flowers, which the plant needs to attract pollinators, from the traps, which would otherwise capture and kill those same pollinators. The flowers are white with green veining, arranged in a cluster at the top of the stalk, and emit a faint, pleasant fragrance. Native pollinators, including various species of small bees and beetles, visit the flowers and transfer pollen between plants, facilitating cross-pollination. After successful pollination, the flowers develop into small capsules filled with tiny black seeds. These seeds are lightweight and can be dispersed by wind or water, though dispersal distances are typically limited to a few feet or meters from the parent plant. The seeds germinate best on bare, moist soil, and seedlings grow slowly, taking 4 to 5 years to reach maturity and begin flowering. This long developmental period makes Venus flytrap populations slow to recover from disturbance and particularly vulnerable to habitat destruction or overharvesting. In their natural wetland habitats, Venus flytraps coexist with other carnivorous plant species, creating communities of nutrient-gathering specialists. The same bog that supports a Venus flytrap population might also host sundews, with their adhesive tentacles glittering in the sun, and pitcher plants with their tall tubular traps filled with rainwater and drowned insects. Each species occupies a slightly different niche, capturing prey of different sizes or at different heights above the ground, reducing direct competition. The Venus flytrap snap trap mechanism is particularly effective for capturing ants, spiders, beetles, and grasshoppers, mobile prey that might escape from the slower, acting adhesive traps of sundews or the passive pitfall traps of pitcher plants. The relationship between Venus flytraps and their prey is not entirely one-sided. While the plant benefits nutritionally from capturing insects, not all insects that visit the traps become victims. Some small insects are too lightweight or too quick to trigger the traps reliably. Others, particularly certain species of spiders, have learned to hunt on the traps themselves, positioning themselves near the traps opening to ambush other insects attracted to the plant's nectar. These spiders benefit from the Venus flytrap's attractiveness to prey without suffering the consequences of triggering the trap, a clever ecological exploitation of the plant's hunting strategy. The biochemistry of the Venus flytrap's electrical signaling system has been studied intensively because it offers insights into fundamental questions about how plants process information and respond to their environment. Plants lack neurons, the specialized cells that transmit electrical signals in animals. Yet they can generate and propagate action potentials that are remarkably similar to the nerve impulses in our own bodies. In both cases, the mechanism involves the movement of charged ions, primarily calcium, potassium, and chloride, across cell membranes through voltage-gated ion channels. When a trigger hair is bent, mechanosensitive ion channels at the base of the hair open in response to the physical stress on the cell membrane. This allows positively charged calcium ions to flood into the cell, changing the electrical potential across the membrane. If enough calcium enters, the membrane potential reaches a threshold that triggers voltage-gated ion channels to open, amplifying the signal and causing it to propagate to neighboring cells through structures called plasmodesmata, tiny channels that connect adjacent plant cells and allow the direct passage of ions and small molecules. The action potential spreads across the trap like a wave, traveling through the leaf tissue in all directions from the point of stimulation. The counting mechanism, the ability of the plant to require 2 stimulations rather than responding to just one, involves a form of short-term memory. The first action potential causes changes in ion concentrations and membrane potentials that persist for several seconds. If a second action potential arrives within this window, typically 20 to 30 seconds, the cumulative effect of both signals exceeds the threshold needed to trigger the trap closure. If too much time passes between stimulations, the cellular conditions reset to their baseline state, and the count effectively restarts at 0. This elegant system allows the plant to distinguish between random environmental stimuli and genuine prey with a level of sophistication that rivals simple neural circuits in animals. The snap of the trap, faster than the blink of an eye, represents one extreme in the spectrum of carnivorous plant strategies. It is active trapping in its most dramatic form, a split-second ambush predator in the botanical world. But as we will see, not all carnivorous plants rely on speed. Some take the opposite approach entirely, using patience and architecture rather than motion and momentum, constructing elaborate passive traps that require no movement whatsoever, yet prove equally effective at securing the nutrients that these remarkable plants need to survive in their challenging habitats. The diversity of solutions that evolution has crafted to solve the same fundamental problem reveals the extraordinary creativity inherent in natural selection, the endless variations on a theme that emerge when life confronts limitation and finds a way forward despite it. Imagine now a different kind of trap altogether, one that requires no sudden movements, no electrical signals racing through plant tissue, no counting of touches or split-second decisions. Picture, instead, a vessel, elegant in its simplicity, waiting with the patience that only a rooted being can possess. This is the pitcher plant, and its strategy is as ancient as it is effective. Not to chase or grab, but simply to offer an irresistible invitation, and then make escape impossible. Pitcher plants belong to several unrelated plant families that have independently evolved the same basic solution to the nutrient problem. The most species-rich families are the Nepenthes, with over 100 species of tropical pitcher plants native to Southeast Asia, and the Sarraceniaceae, containing the North American pitcher plants that grow in the bogs and wetlands of the eastern United States and southeastern Canada. Despite their evolutionary separation by more than 100 million years, these plants have converged on remarkably similar designs, testament to the power of natural selection when faced with identical challenges. Consider the architecture of a Nepenthes pitcher, one of those tropical species that dangle from tendrils extending from the tips of ordinary leaves. The pitcher itself is a highly modified leaf structure, though it looks nothing like the flat, photosynthetic surfaces we associate with foliage. It hangs like a small vase, or urn, sometimes brightly colored in reds and purples and greens, sometimes mottled with patterns that resemble flowers, or perhaps rotting organic matter. Visual signals calibrated to attract the particular insects that thrive in the plant's habitat. The opening of the pitcher is crowned by a lid, held aloft on a hinge, positioned to shelter the interior from excessive rainfall that might dilute the digestive fluids or cause the pitcher to overflow and lose its accumulated prey. The rim of the pitcher, called the peristome, is one of nature's most sophisticated surfaces. In many Nepenthes species, it is sculpted into a series of ridges that radiate inward like the threads of a screw. Each ridge slick with nectar that attracts ants, flies, beetles, and other crawling insects. The peristome's secret lies not just in its shape, but in its wettability under humid conditions, which are nearly constant in tropical rainforests. The peristome becomes coated with a thin film of condensation or nectar. This water film is critical because it enables a phenomenon called aquaplaning. When an insect steps onto the wet peristome in search of the nectar, concentrated in specialized glands along the rim, its feet cannot gain purchase. The thin layer of water prevents the insect's adhesive pads or claws from making contact with the solid surface beneath. The insect slips, and once it begins to slide on the angled, inward, curving peristome, there is no recovery. Gravity completes what the slippery surface began. Below the peristome, the inner wall of the pitcher is divided into distinct zones, each with its own function in the trapping process. The upper portion of many Nepenthes species is coated with a layer of microscopic waxy crystals that project perpendicularly from the surface, like a forest of tiny needles. These crystals are structurally fragile, designed to break off at the slightest contact. When an insect that has survived its tumble from the peristome attempts to climb back up the pitcher wall, its feet contact these wax crystals. The crystals snap away from the surface, contaminating the insect's adhesive pads and claws, rendering them useless. It is as if the insect is trying to climb a wall coated in ball bearings. Each step dislodges more crystals, and the insect slides inexorably downward toward the pool of liquid waiting at the pitcher's base. Some pitcher plant species lack the waxy coating. Recent research has revealed an additional mechanism at work. The digestive fluid itself possesses remarkable physical properties. In species like Nepenthes rafflesiana, the fluid at the bottom of the pitcher is not simply a watery solution, but a highly viscoelastic substance, meaning it is both viscous like honey and elastic like rubber. When an insect falls into this fluid and attempts to swim or crawl out, the fluid's elasticity causes it to stick to the insect's body in long, stretchy filaments. These filaments act like countless tiny ropes, pulling the insect back down each time it tries to escape. The struggling insect becomes progressively more entangled, its movements growing weaker as exhaustion sets in, until finally it drowns in the very fluid that will soon dissolve its tissues. The liquid pool at the base of a Nepenthes pitcher is itself a complex ecosystem. In unopened pitchers, the plant secretes a digestive fluid that contains enzymes similar to those produced by the Venus flytrap, proteases to digest proteins, chitinases to break down exoskeletons, phosphatases to liberate phosphorus. The fluid is typically acidic, with a pH that can range from highly acidic in some species to nearly neutral in others, depending on the species' particular digestive strategy. As the pitcher ages and begins to capture prey, the fluid's composition changes. Bacteria colonize the pitcher, washing in with rainwater, or arriving on the bodies of captured insects, These bacterial communities contribute additional digestive enzymes, and in some species, they appear to be more important for breaking down prey than the plant's own secretions. In certain Nepenthes species, particularly those with less acidic fluids, nitrogen fixing bacteria take up residence in the pitcher liquid. These bacteria can capture atmospheric nitrogen and convert it into forms the plant can use, providing yet another pathway for the plant to acquire this essential nutrient. The relationship between pitcher plants and their bacterial inhabitants represents a fascinating example of mutualism. The plant provides a habitat and a steady supply of organic matter in the form of drowned insects, while the bacteria produce enzymes and fixed nitrogen that benefit the plant. The pitcher becomes not just a trap, but a miniature aquatic ecosystem, complete with its own food web of bacteria, protists, and in some cases even mosquito larvae and other insects that have adapted to live within the deadly fluid without being digested themselves. North American pitcher plants, the Sarracenia species, employ a similar but distinctly different strategy. Unlike their tropical cousins, which dangle pitchers from tendrils, Sarracenia plants grow as rosettes of upright pitchers emerging directly from the ground. Each pitcher is a complete leaf that has evolved into a hollow tube, often vividly colored and patterned, rising anywhere from a few inches to more than 3 feet tall, depending on the species. The opening of the pitcher faces upward or slightly outward, easily accessible to flying and crawling insects attracted by the plant's visual signals and the sweet scent of nectar produced by glands around the pitchers. Rim and on the hood, a leaf structure that arches over the opening in most species. The hood serves multiple functions. In most Sarracenia species, it prevents rainwater from excessively diluting the digestive fluid inside the pitcher, though in the purple pitcher plant, Sarracenia purpurea, the hood is reduced, and the pitcher fills with rainwater, creating a different kind of trap that relies more on bacterial digestion than on plant-produced enzymes. The hood also bears nectar glands that continue to lure insects even after they have landed on the pitcher. An ant following a nectar trail might walk along the hood's underside, then onto the pitcher's rim, then venture inside the pitcher opening in search of more nectar, unaware that it is descending into a trap from which there is no return. The interior of a Sarracenia pitcher is divided into several functional zones. Just below the opening is a zone where the inner surface is relatively smooth and may still bear nectar glands, continuing to draw the insect deeper into the tube. Further down, the surface becomes covered with downward pointing hairs, stiff bristles that allow an insect to walk easily downward, but make upward movement nearly impossible. These hairs act like a ratchet, ensuring that any insect that enters the lower portions of the pitcher cannot climb back out. In some species, particularly the cobra lily Darlingtonia californica, the pitcher has evolved translucent patches in its walls, creating false exits that confuse trapped insects. The insect, seeking the light and believing it has found an escape route, flies or crawls toward these windows, like aerials, only to bounce off the pitcher wall and fall deeper into the trap. At the bottom of the pitcher lies a pool of liquid containing digestive enzymes, though in many Sarracenia species, this fluid is less acidic and enzyme-rich than in Nepenthes. The purple pitcher plant, for instance, relies heavily on its community of bacteria and other microorganisms to break down captured prey. Over the course of a growing season, a single Sarracenia pitcher might accumulate dozens or even hundreds of insects, creating a nutrient-rich soup that the plant absorbs through specialized glands on the lower inner wall of the pitcher. The exoskeletons of the prey, which cannot be fully digested, accumulate at the bottom of the pitcher, gradually filling it with a dark mass of chitin and other indigestible material. By the end of the season, when the pitcher dies back, it releases this accumulated organic matter into the soil, enriching the immediate environment around the plant. The life cycle of a pitcher is itself a study in resource allocation. Producing a pitcher requires significant investment of energy and nutrients. The plant must construct not just the pitcher structure itself, but also the specialized glands for nectar production and digestive enzyme secretion. Each pitcher functions for a single growing season, from spring through autumn in temperate species, or year-round in tropical species that produce pitchers continuously. Over this period, a successful pitcher might capture enough prey to more than repay the plant's investment, providing nitrogen and phosphorus that fuel the production of new pitchers and support the plant's reproductive efforts. The flowering stalks of pitcher plants are particularly striking, rising high above the pitchers on tall stems that can reach 2 or 3 feet in some species. This elevation serves an important purpose. It separates the flowers, which must attract pollinators, from the pitchers, which would kill those same pollinators if they ventured too close. The flowers of Sarracenia are complex and beautiful, with elaborate structures that ensure cross-pollination. The 5-petaled flowers hang downward like umbrellas, and visiting bees must crawl underneath the flower's unusual umbrella, like style picking up pollen from the dangling anthens, before moving to another flower to deposit that pollen on the receptive stigma surfaces. This intricate floral architecture prevents self-pollination and encourages genetic diversity within pitcher plant populations. The ecological role of pitcher plants extends beyond their individual nutrient acquisition. In the nutrient-poor wetlands where they grow, pitcher plants help concentrate nitrogen and other nutrients, creating localized hotspots of fertility. The prey they capture represents a transfer of nutrients from the broader landscape into the bog ecosystem. Insects, which feed on plants across a wide area, carry those nutrients into the bog when they visit pitcher plants and become trapped. The pitcher plant then releases those nutrients into the local environment, both through its own eventual decay and through the absorption and incorporation of nutrients into its tissues, which other organisms may later consume. Pitcher plants also provide habitat for a remarkable array of specialized organisms. Certain species of mosquitoes lay their eggs exclusively in pitcher plant fluids, and their larvae have evolved immunity to the digestive enzymes and low oxygen levels that would kill most aquatic insects. These mosquito larvae feed on the smaller organisms in the pitcher fluid and on particles of decomposing prey, and in turn become food for larger predators that also inhabit the pitchers. Some spiders build webs across the openings of pitchers, capturing insects attracted to the plant's nectar before they can enter the trap. A few species of tree frogs use Nepenthes pitchers as breeding pools, laying their eggs in the fluid where the tadpoles develop, protected from most predators, but feeding on the nutrient-rich environment created by the plant's captures. Let your attention shift now from these architectural marvels, these patient vessels of capture, to yet another approach, one that combines elements of both passive patience and active response. The Sundews, members of the genus Drosera, have evolved a strategy that bridges the gap between the immobile pitcher and the explosive snap trap. They are flypaper plants, relying on adhesive rather than pitfalls. Yet many species can move their leaves and tentacles in response to captured prey, maximizing contact and ensuring that their victims cannot escape. Picture a sundew in your mind. If you can, it grows as a low rosette of leaves, often in the same nutrient, poor bogs and wetlands where pitcher plants and Venus flytraps thrive. Each leaf is adorned with hundreds of hair, like structures called tentacles, though they are not tentacles in the animal sense, but rather specialized plant hairs, technically called trichomes, each topped with a gland that secretes a droplet of clear, glistening fluid. In morning light or after a rain, these droplets catch the sun and sparkle like dewdrops, hence the common name sundew, though the liquid is anything but water. The mucilage produced by sundew glands is a remarkable substance, a viscoelastic biopolymer with properties that make it extraordinarily effective as an insect trap. It is sticky, far stickier than water, forming strong adhesive bonds with the waxy cuticles of insect bodies. It is viscous, resisting flow and making it difficult for an insect to pull free. And most remarkably, it is elastic, capable of stretching to nearly 1,000,000 times its original size without breaking. When a small fly lands on a sundew leaf and contacts one of these mucilage droplets, the fluid instantly adheres to the insect's body. As the fly struggles to escape, trying to pull its legs free, the mucilage stretches into long, thin filaments that remain attached both to the insect and to the plant. More tentacles come into contact with the struggling insect, each adding its own droplet of adhesive fluid, and soon the fly is hopelessly entangled in a web of elastic threads, its wings stuck, its legs immobilized, its movements growing weaker as exhaustion and suffocation set in. The mucilage itself is not merely a passive trap. It contains digestive enzymes that begin working even before the insect dies. Proteases start breaking down proteins in the insect's cuticle and body tissues. The mucilage is acidic, typically around pH 5, which helps the enzymes function efficiently and may also contribute to the insect's demise by damaging its tissues. The prey typically dies within 15 minutes of being caught, either from exhaustion, from suffocation as the mucilage blocks its spiracles, the breathing holes along its body, or from the direct effects of the digestive enzymes on its tissues. But the sundew does not rely solely on the initial capture. Many species exhibit remarkable movement in response to trapped prey. When an insect becomes stuck and begins struggling, the mechanical stimulation and chemical signals from the prey trigger a response in the plant. The tentacles that have not yet touched the prey begin to bend slowly inward, curling toward the center of the leaf where the insect is trapped. This bending is driven by differential growth rates on opposite sides of the tentacle stalk, a process mediated by the plant hormone jasminate, and by changes in cell turgor pressure. It takes minutes to hours for the tentacles to complete their inward bend. But the result is that more and more tentacles make contact with the prey, maximizing the surface area over which digestion can occur and ensuring that the insect is completely enveloped in digestive mucilage. In some sundew species, The leaf blade itself participates in this movement. The entire leaf slowly curves upward along its edges, forming a shallow cup that further entraps the prey. This leaf movement can take hours to complete, and it appears to occur only when the plant detects both mechanical stimulation and chemical signals, indicating genuine prey rather than debris. The plant is, in effect, assessing what it has caught and responding accordingly. The chemical composition of the mucilage is a subject of ongoing research. It consists primarily of long-chain polysaccharides, complex sugar molecules that form the sticky, elastic matrix. Dissolved within this matrix are digestive enzymes, including proteases, esterases, peroxidases, and phosphatases. The fluid also contains ions, particularly calcium, magnesium, potassium, and sodium, which may play roles in maintaining the mucilage's physical properties and in facilitating enzyme activity. Some researchers have found that the mucilage contains networks of nanofibers and nanoparticles, visible only under electron microscopy, structures that may contribute to the fluid's remarkable stickiness and elasticity. The genus Drosera is one of the largest groups of carnivorous plants, with nearly 200 species distributed across every continent except Antarctica. They range in size from tiny species with leaves smaller than a fingernail to the king sundew of South Africa, whose leaves can reach more than a foot in length. Some species are tropical and grow year-round, while others are temperate and die back to dormant buds or tubers during cold winters. Some species have evolved specialized trap structures beyond the standard sticky leaves. The Australian sundew Drosera glanduligera, for instance, has evolved two types of tentacles: sticky glue tentacles in the center of the leaf, and specialized snap tentacles around the leaf's periphery. These snap tentacles lack mucilage, but can bend explosively fast, in as little as 75 milliseconds, catapulting any insect that touches them into the sticky tentacles at the leaf's center, combining the speed of active trapping with the reliability of adhesive capture. Once a sundew has captured and killed its prey, digestion proceeds over several days to several weeks, depending on the size of the prey and environmental conditions such as temperature. The enzymes in the mucilage and those secreted by sessile glands on the leaf surface work to break down proteins, fats, and nucleic acids. The nutrients released by this digestion are absorbed through the leaf surface, primarily through the sessile glands that are scattered among the stalked tentacles. These sessile glands lack the tall stalks and mucilage droplets of the tentacles, but they are the primary sites of nutrient absorption, shuttling nitrogen, phosphorus, and other minerals from the external environment into the plant's vascular system. When digestion is complete, the tentacles slowly return to their upright position, ready to trap new prey. The dried husk of the insect, typically just the empty exoskeleton, and any wings or other indigestible parts, may remain stuck to the leaf for a while before being washed away by rain or blown away by wind. A single sundew leaf can capture multiple insects over its lifetime, which typically lasts one growing season before the leaf dies and is replaced by new growth from the plant center. The Sundews combination of passive adhesive trapping and active tentacle movement represents an intermediate strategy between the purely passive pitchers and the purely active snap traps. Like pitcher plants, Sundews rely on lures and structural adaptations to capture prey without explosive movement. Like Venus flytraps, they can sense prey and respond with movement, though their motion is measured in minutes rather than milliseconds. This intermediate strategy has proven highly successful, allowing Sundews to colonize a wide range of habitats, from Arctic bogs to tropical rainforests to Mediterranean heathlands. The study of sundew movement contributed significantly to our understanding of plant behavior. Charles Darwin was fascinated by sundews, conducting extensive experiments on the common sundew Drosera rotundifolia, and devoting a substantial portion of his book on insectivorous plants to these remarkable organisms. He wrote that he cared more about Drosera than about the origin of all the species in the world, a testament to his wonder at these plants' almost animal-like responsiveness. His careful observations documented how mechanical stimulation could trigger tentacle movement, how the plant could distinguish between nutritious prey and inedible objects, and how the speed and extent of the response depended on the nature of the stimulus. Modern research has revealed the mechanisms underlying Darwin's observations. When a tentacle is stimulated by trapped prey, the plant generates electrical signals similar to action potentials that propagate to neighboring tentacles, triggering their bending response. The signaling involves the same ion channels and similar electrochemical changes that occur in the Venus flytrap, though the response is much slower. Chemical signals, particularly the hormone jasminate, also play a crucial role, integrating information about the mechanical stimulation and the chemical composition of whatever has been caught. The plant's ability to integrate multiple signals and produce a coordinated, appropriate response demonstrates a level of sophistication that challenges simple distinctions between plants and animals, between passive and active, between simple and complex. The nutritional benefits that sundews derive from carnivory are substantial and measurable. Experimental studies have shown that sundews fed regularly with insects exhibit markedly increased growth rates, produce more leaves, and flower more abundantly than unfed plants, growing in the same conditions. The nitrogen obtained from digested prey is incorporated directly into proteins, nucleic acids, and chlorophyll, enhancing the plant's photosynthetic capacity, even as the carnivorous leaves themselves are somewhat compromised in their ability to photosynthesize efficiently. This creates an interesting feedback loop. Carnivory provides nutrients that enable the plant to produce more carnivorous leaves, which capture more prey, which provides more nutrients. In optimal conditions, a well-fed sundew can achieve growth rates that rival non-carnivorous plants growing in nutrient-rich soils. The diversity within the sundew genus extends beyond simple variations in size and habitat. Some species have evolved underground tubers that allow them to survive harsh, dry seasons, dying back completely above ground and waiting out months of heat and drought before resprouting when rains return. Others produce specialized winter buds called hibernacula, dense rosettes of overlapping leaves that protect the growing point through freezing temperatures. Still others are tuberous species that are active during winter and dormant during summer. reversing the typical pattern of temperate plants. This diversity reflects the genus' ancient origins and its wide distribution across varied climates and ecosystems. Related to the sundews, but employing a slightly different strategy, are the Butterworts, plants of the genus Pinguicula. These carnivorous plants might seem unremarkable at first glance, appearing as simple rosettes of yellowish-green butter, colored leaves pressed close to the ground. The leaves feel greasy to the touch, as if coated with some oily substance, and indeed, the common name Butterwort derives from this greasy texture. But this innocent appearance conceals an effective trapping mechanism that has allowed Butterworts to colonize habitats from Arctic tundra to tropical highlands. The upper surface of a Butterwort leaf is covered with two types of glands. Stalked glands produce a mucilaginous secretion, similar to that of sundews, though typically less elastic and more viscous, forming a thin coating across the entire leaf surface rather than discrete droplets. Sessile glands flush with the leaf surface, secrete digestive enzymes once prey has been captured. The mucilage is not as visually conspicuous as the glistening dewdrops of sundews, but it is highly effective at trapping small insects, particularly fungus gnats, fruit flies, and other tiny flying insects that land on the leaf surface. Once an insect becomes stuck in the mucilage, Butterworts in many species respond with a slow curling of the leaf margins. The edges of the leaf gradually roll upward and inward over the course of hours or days, bringing more of the leaf's digestive glands into contact with the prey and creating a shallow digestive cavity that concentrates the enzymes and prevents rain from washing away the partially digested remains. This curling is less dramatic than the tentacle movements of sundews or the snap of Venus flytraps, but it serves a similar function, maximizing the plant's contact with its prey and optimizing the conditions for digestion. The ecology of Butterworts is fascinating in its own right. Many species grow on wet rock faces, cliffs, and seepage areas where water constantly trickles over mineral, poor substrates. The plants root in thin accumulations of moss and organic debris, extracting what little nutrients they can from this meager soil while supplementing their diet with the insects that are attracted to the moisture and shade of these seepage habitats. Some Butterwort species have evolved highly specialized relationships with their habitats. Certain Mexican species, for instance, grow on vertical limestone cliffs where they are bathed in fine spray from waterfalls, their leaves forming tight rosettes that prevent them from being washed away, while still allowing them to capture tiny insects from the mist-laden air. Butterworts exhibit remarkable seasonal plasticity in many species. During the growing season, when insects are abundant, the plants produce carnivorous leaves designed for trapping prey. But as autumn approaches and insect populations decline, some species transition to producing non-carnivorous winter leaves, succulent structures that lack the sticky glands but are better adapted for surviving cold temperatures. This seasonal dimorphism allows the plants to optimize their form for the resources available at different times of year. The genus Pinguicula contains over 125 currently accepted species, distributed primarily in the Northern Hemisphere, with centers of diversity in Mexico and Central America, and with smaller numbers found in Europe, North America, and Northern Asia. Like sundews, Butterworts show remarkable variation in form and adaptation, despite their relatively simple leaf structure. Some species are tiny, with leaves smaller than a dime, while others produce leaves several inches long. Some are adapted to freezing temperatures and grow in alpine bogs, while others thrive in warm, humid, tropical cloud forests. This diversity reflects millions of years of evolution and adaptation to local conditions, each species fine-tuning its carnivorous strategy to the particular insects and environmental conditions it encounters. The digestive enzymes produced by butterworts are similar to those of other carnivorous plants, but with some interesting variations. Proteases break down insect proteins into amino acids. Phosphatases liberate phosphorus from organic molecules in the prey's tissues. Some species also produce chitinase, the enzyme that breaks down the tough chitin in insect exoskeletons, though butterworts generally capture smaller, softer-bodied prey than plants like Venus flytraps or pitcher plants. So chitinase may be less critical to their digestive process. The digestive fluid produced by butterworts is mildly acidic, creating conditions favorable for enzyme activity, while also potentially weakening the prey's exoskeleton and making it more permeable to the digestive enzymes. The absorption of nutrients from digested prey in butterworts occurs primarily through the sessile glands scattered across the leaf surface. These glands are capable of taking up amino acids, ammonium ions, nitrate, phosphate, and other nutrients released during digestion. The absorbed nutrients are then transported through the leaf tissue into the plant's vascular system and distributed throughout the plant. Studies using isotopically labeled prey have shown that nitrogen from captured insects can be detected throughout the plant within hours of capture, demonstrating the efficiency of the absorption process. One particularly interesting aspect of butterwort ecology is the potential for these plants to trap their own pollinators. Like other carnivorous plants, butterworts elevate their flowers well above their leaves on tall stems, physically separating the attractive flowers from the deadly leaves. But research has shown that some butterwort species do occasionally capture pollinating insects that, after visiting the flowers, land on the leaves below. This represents a cost to the plant's reproductive success, as each pollinator captured is one fewer insect available to transfer pollen between flowers. Natural selection appears to have struck a balance, with the flower stalks tall enough to reduce pollinator capture to acceptable levels, while still connecting the flowers to the plant's nutrient transport system. The evolution of carnivory in plants like sundews, butterworts, and pitcher plants reveals a fundamental principle of biology. Organisms can evolve similar solutions to similar problems. Despite having vastly different evolutionary histories, the adhesive traps of sundews and butterworts evolved independently from one another, and from the pitfall traps of pitcher plants. Yet, all three strategies achieve the same end result: supplementing the plant's nutrition with animal-derived nutrients in environments where soil nutrients are scarce. The specific details differ. The molecular mechanisms have distinct origins, but the functional outcome is remarkably similar. This convergent evolution extends even to the molecular level. When researchers examined the digestive enzymes of different carnivorous plant families, they found that while the enzymes themselves had evolved from different ancestral genes, natural selection had acted on similar genetic pathways in each lineage. Genes involved in defense against pathogens were repurposed for digesting prey. Genes that normally function in roots were expressed in leaves. Transport proteins that typically move nutrients from soil were deployed to absorb nutrients from the external surface of traps. Evolution worked with the genetic raw materials available in each plant lineage, modifying existing genes rather than creating entirely new ones, yet arriving at functionally similar solutions. The study of carnivorous plants has implications that extend far beyond botanical curiosity. The adhesive properties of sundew mucilage, for instance, have attracted interest from materials scientists and biomedical researchers. The mucilage's combination of strong adhesion, elasticity, and biocompatibility suggests potential applications in wound treatment, surgical adhesives, and tissue engineering. When dried, the mucilage provides a suitable substrate for the attachment and growth of living cells, opening possibilities for using carnivorous plant materials in regenerative medicine or as coatings for medical implants that could improve integration with surrounding tissue and reduce the risk of rejection. The digestive enzymes produced by carnivorous plants are also subjects of ongoing research. Understanding how these plants produce and regulate enzyme secretion in response to prey capture could inform agricultural biotechnology, potentially leading to crops that are more efficient at extracting nutrients from organic matter in the soil. The signaling pathways that allow carnivorous plants to distinguish between nutritious prey and inedible debris might inspire sensors or smart materials that can detect and respond to specific chemical signatures. From an ecological perspective, carnivorous plants serve as model systems for studying nutrient cycling, plant-animal interactions, and adaptation to extreme environments. The bogs and wetlands where many carnivorous plants grow are among the most nutrient-poor habitats on Earth. Yet, they support diverse communities of specialized organisms that have evolved remarkable strategies for coping with scarcity. Carnivorous plants are keystone species in many of these ecosystems, concentrating nutrients through their predation and making those nutrients available to other organisms through their own growth, reproduction, and eventual decay. As you contemplate these plants, these patient hunters with their elegant traps and sophisticated chemistry, consider the larger story they tell about life's adaptability. In environments that would seem to preclude plant growth, where soil offers almost nothing and survival should be impossible, evolution has crafted solutions of stunning ingenuity. Leaves have become pitchers, glands have become sticky traps, defensive enzymes have become digestive systems, and the boundary between plant and predator has blurred in ways that continue to fascinate and instruct us. The diversity of these solutions, from the explosive snap of the Venus flytrap to the patient architecture of the pitcher to the sticky embrace of the sundew, reveals the creative power inherent in natural selection when life confronts limitation and finds not one path forward, but many. Among the fastest movements ever recorded in the plant kingdom, faster even than the Venus flytrap's 100 millisecond snap, are the traps of the Bladderworts. The genus Utricularia, containing approximately 233 species, distributed across every continent except Antarctica, has evolved perhaps the most sophisticated carnivorous mechanism in the entire plant world. These are plants that have mastered the physics of suction at scales so small and speeds so fast that they challenge our understanding of what plants can do. You've felt the sudden pull of suction before. Bladderworts are aquatic or semi-aquatic plants that grow in freshwater, ponds, lakes, bogs, and waterlogged soils. Many species are free, floating, drifting through still or slow, moving water with no roots to anchor them, their entire structure dedicated to photosynthesis and prey capture. Others live terrestrially in saturated soils. Their tiny traps hidden underground, where they hunt in the microscopic realm between soil particles. What makes bladderworts unique among all carnivorous plants is the bladder itself, a hollow underwater suction cup, a trap that works not through sticky adhesives or digestive pitchers or snapping lobes, but through the sudden violent influx of water driven by a pressure differential. Imagine a small, hollow sphere, translucent and delicate, only a few millimeters across at most. The bladder walls are thin and flexible, but strong enough to maintain their shape under stress. Through active pumping, the plant continuously moves water out of the bladder's interior, creating a partial vacuum inside, a region of lower pressure than the surrounding environment. The bladder walls, stressed by this pressure difference, deform inward, storing elastic energy like a compressed spring. At the entrance to this bladder sits a door, a trap door that seals the opening watertight, held shut by the pressure difference. On the outer surface of this door are trigger hairs, fine bristles that extend into the water, sensitive to the slightest touch. When a small aquatic organism, perhaps a water flea, a copepod, a mosquito larva, a tiny worm, swims near the bladder and brushes against one of these trigger hairs, the door responds instantaneously. In some species of Utricularia, the door opens in as little as 300 to 700 microseconds, less than 1,000th of a second, a time frame so brief that it ranks among the fastest movements known in the plant kingdom, faster than the blink of an eye by 2 orders of magnitude. The opening itself is a mechanical marvel. The door, which in its resting state is curved outward, suddenly inverts its curvature through a process called buckling, flipping from convex to concave in an instant. This buckling is not a consequence of the suction, but rather the trigger for it. The door's geometry is carefully tuned, so that when the trigger hairs are stimulated, the pressure differential reaches a critical threshold, and the door buckles inward. Opening the trap, the moment the seal breaks, water rushes into the bladder with tremendous force. The sudden influx is driven by the pressure difference between the bladder's interior and the surrounding water. And because the opening happens so quickly, the flow is dominated by inertia rather than viscosity. Water accelerates inward at speeds reaching up to 4 meters per second econd, carrying with it anything in its path. The prey organism, caught in this torrent, is swept into the bladder in a fraction of a millisecond. Researchers have measured the acceleration experienced by captured prey at up to 600 times the force of gravity, an acceleration so extreme that it leaves the animal with virtually no chance of escape. Even organisms that might normally be capable of rapid swimming responses are unable to react in time. The suction time scales outpace the sensoromotor responses of most aquatic invertebrates. By the time the animal's nervous system registers the flow, it has already been pulled inside, and the door has snapped shut behind it. The entire capture event, from door opening to door closing, takes between 10 and 15 milliseconds in most species. Once the door closes, the bladder is sealed again, and the prey is trapped inside. The interior of the bladder is lined with glands, quadruped glands that have 4 arms radiating from a central stalk. And these glands immediately begin secreting digestive enzymes. Proteases, phosphatases, and other enzymes break down the soft tissues of the captured organism. Over the course of hours or days, nutrients from the prey, particularly nitrogen and phosphorus, are absorbed through the bladder walls and transported throughout the plant. After digestion is complete, the bladder resets itself. Water is pumped out once more. The pressure differential builds. The door regains its outward curvature, and the trap is ready to fire again. This reset process takes approximately 20 to 30 minutes. And then the bladder is once again primed, waiting for the next touch of a trigger hair. Some bladderworts, in the absence of prey, will fire spontaneously after several hours. These spontaneous firings may serve to capture microscopic algae or phytoplankton that wouldn't otherwise trigger the trap, giving the plant an omnivorous diet. The traps can fire hundreds or even thousands of times over the life of the plant, making bladderworts extraordinarily efficient predators in nutrient-poor aquatic environments. The diversity within the genus Utricularia is remarkable, spanning aquatic, terrestrial, and even epiphytic lifestyles across nearly every corner of the world. Aquatic species like Utricularia vulgaris, the common bladderwort, float freely in lakes and ponds across the northern hemisphere, their stems bearing numerous bladders and their bright yellow flowers rising above the water surface on slender stalks. These aquatic forms create dense underwater meadows, their branching stems intertwining to form mats that drift through the water column. Each plant may bear dozens or even hundreds of bladders, creating a floating predatory network that can significantly impact zooplankton populations in the waters where they grow. The flowers themselves are often strikingly beautiful, bilateral blooms in shades of yellow, purple, white, or pink, held above the water on stems that can reach 15 to 20 centimeters tall, ensuring that pollinators, typically bees and other flying insects, are kept well away from the deadly traps below. Terrestrial species inhabit a different world entirely. Found in the wet soils of bogs, fens, tropical savannas, and sandy seeps, these bladderworts produce tiny bladders less than a millimeter in diameter, sometimes as small as 0.2 millimeters, invisible to the naked eye but perfectly sized for hunting protozoa and rotifers in the thin film of water that coats soil particles. These microscopic hunters live in the spaces between grains of sand or peat, their traps set in the dark, saturated soil where light never reaches. The terrestrial bladderworts often form small rosettes of leaves at the soil surface, green structures that photosynthesize while the subterranean traps do the work of capturing prey. Some species in seasonally dry climates have adapted to produce tubers or other storage organs, allowing them to survive periods of drought when the soil dries out and prey becomes unavailable. Then there are the epiphytic species, bladderworts that have taken to the trees. In the humid tropical forests of South America, Africa, and Asia, certain Utricularia species grow on the branches and trunks of trees, their roots anchoring them to bark. Their bladders suspended in the small pools of water that collect in the axils of leaves or in the crevices of moss. These aerial bladderworts hunt in miniature aquatic ecosystems high above the forest floor, capturing mosquito larvae, algae, and protozoa that colonize these ephemeral water bodies. The epiphytic lifestyle is precarious, dependent on regular rainfall to maintain the water supply that the plants and their prey require, but it allows the bladderworts to escape competition from terrestrial and aquatic carnivores and to access prey resources that few other plants can exploit. The range of bladder sizes across the genus reflects this ecological diversity and the range of prey sizes available in different habitats. The smallest bladders, measuring just 0.2 millimeters across, target single-celled organisms like paramecia and euglenoids. Creatures so small they can pass through the eye of a needle. The largest bladders, reaching 1.2 centimeters in length, in some tropical species, can capture substantial prey, including water fleas, several millimeters long, mosquito larvae, nematodes, and even tadpoles and small fish fry. This size range represents a 60-fold variation in linear dimension and an even greater variation in volume. Yet all of these traps, from the tiniest to the largest, operate on the same fundamental principle: the sudden release of stored elastic energy to generate rapid suction. The fact that this mechanism scales so effectively across such a range of sizes is a testament to its robustness and to the evolutionary success of the bladderwort lineage. The physics of bladderwort traps has fascinated researchers for decades. At the small scales where these traps operate, viscosity normally dominates fluid flow. In the so-called creeping flow regime, thick, syrupy forces prevent the generation of fast, efficient suction. A suction-feeding animal as small as a bladderwort trap would struggle to capture prey because the water would resist flowing quickly enough. Yet bladderworts escape this limitation through two key adaptations. First, they generate extraordinarily strong suction pressures by pumping water out of the bladder so thoroughly and by maintaining such a significant pressure differential. They ensure that when the door opens, the force driving the inflow is powerful enough to overcome viscous resistance. Second, they open the door so quickly that the flow reaches peak velocity almost instantaneously. This sudden onset means the flow is inertial from the very beginning, with water accelerating before viscosity has a chance to slow it down. The result is a suction mechanism that works at scales where suction should not work, a testament to the evolutionary fine-tuning of these remarkable structures. Bladderworts also demonstrate the concept of entrance effects in fluid dynamics. When water enters the bladder through the narrow opening, the flow does not immediately settle into a smooth, fully developed pattern. Instead, it remains undeveloped near the entrance, maintaining higher velocities than would be possible in a fully developed flow. This entrance effect further contributes to the speed and effectiveness of the trap. The morphological demands of generating such rapid, powerful suction are exacting. The bladder walls must be thin enough to flex and store elastic energy, yet strong enough to withstand the repeated stress of thousands of trap cycles. The door must be flexible enough to buckle rapidly, yet robust enough to seal tightly and prevent leakage. The trigger hairs must be sensitive enough to respond to the lightest touch, yet sturdy enough not to break. The glands inside the bladder must be able to secrete enzymes efficiently and absorb nutrients effectively. Every aspect of the trap's anatomy is optimized for its function. the result of millions of years of evolutionary refinement. Bladderworts are not the only aquatic carnivores with rapid movements. Closely related to the bladderworts, and sharing a common ancestor, is a single extraordinary species called the waterwheel plant. Aldrovanda vesiculosa, the sole surviving member of its genus, is the aquatic counterpart of the Venus flytrap, and indeed the two plants are phylogenetic sisters, their most recent common ancestor having lived tens of millions of years ago. Like the Venus flytrap, Aldrovanda possesses snap traps, modified leaves that close around prey in a rapid, violent motion. But unlike the Venus flytrap, which lives on solid ground and hunts flying insects, Aldrovanda drifts freely through the water, rootless and elegant, a living wheel of traps that hunts zooplankton in the slow currents of shallow wetlands. The waterwheel plant is a slender, floating stem, typically 6 to 40 centimeters long, bearing whorls of traps arranged like spokes around the central axis. Each whorl contains 5 to 9 traps, and new whorls are produced regularly as the plant grows, sometimes forming a new whorl every day under optimal conditions. The traps themselves are tiny, only 3 to 5 millimeters long, and they are held aloft by petioles equipped with air sacs that help keep the plant buoyant. One end of the stem grows continuously, while the other end dies back, so the plant is constantly renewing itself, a moving, growing structure that drifts through the water with a slow, graceful motion. The traps are positioned so their openings face outward from the stem, maximizing the chance that a passing organism will swim into their deadly embrace. Each trap consists of 2 lobes, hinged along a midrib, lined on the inside with trigger hairs and digestive glands. When an unsuspecting water flea or mosquito larva brushes against the trigger hairs, the lobes snap shut. The closure is astonishingly fast, taking only 10 to 20 milliseconds, 10 times faster than the Venus flytrap's already impressive 100-millisecond closure. This makes Aldrovanda one of the fastest moving plants on Earth. The mechanism of closure is similar to that of the Venus flytrap, involving a combination of hydraulic pressure changes and the release of prestress stored in the trap lobes. In the open state, the lobes are convex, curved outward. When triggered, they undergo a rapid flip to a concave state, forming a cavity that encloses the prey. The speed of this motion is critical. Underwater, viscous forces resist movement, and a slow closure would give the prey time to swim away. By snapping shut in just 10 to 20 milliseconds, Aldrovanda ensures that even fast-swimming organisms are caught. After the initial closure, the trap continues to tighten. Over the next few minutes to hours, the lobes move closer together, expelling water from the trap's interior and bringing the inner surfaces into contact with the prey. This creates a sealed chamber, a stomach in which digestion takes place. The glands on the inner surface of the lobes secrete enzymes that break down the prey's tissues, and nutrients are absorbed through the trap's epidermis. Each trap can digest 2 to 4 meals before it senesces, and because the plant produces new traps continuously, it maintains a fresh arsenal of functional traps throughout its life. The waterwheel plant is a marvel of efficiency, a carnivore that has adapted to hunt in an environment where movement is constrained by the density and viscosity of water, where prey is small and agile, and where success depends on speed, precision, and relentless predation. Aldrovanda vesiculosa has a wide but fragmented distribution, found in Europe, Asia, Africa, and Australia, though it has disappeared from much of its historical range. It is now considered endangered, with fewer than 50 known populations remaining worldwide, many of them small and isolated. 3/4 of these populations are located in the Chernobyl exclusion zone, a region where human activity has ceased and wetlands have been left undisturbed. Elsewhere, the waterwheel plant faces threats from habitat degradation, eutrophication, water pollution, and the draining of wetlands. It is highly sensitive to changes in water chemistry and even minor shifts in nutrient levels, or pH, can lead to local extinctions. Conservation efforts are underway to protect remaining populations and to reintroduce the plant to areas where it has disappeared. But the future of Aldrovanda remains uncertain. Like many carnivorous plants, it is a sentinel, an indicator of ecosystem health, and its decline signals broader problems in freshwater habitats around the world. Beneath the soil, hidden from view, another group of carnivorous plants pursues prey in the dark, wet underworld of saturated earth. The genus Genlisea, commonly known as corkscrew plants, consists of approximately 30 species distributed across tropical Africa, Central America, and South America. Like bladderworts, to which they are closely related, corkscrew plants are rootless. In place of roots, they have evolved highly specialized underground structures called rhizophils, modified leaves that serve both to anchor the plant and to capture prey. These rhizophils are among the strangest and least understood carnivorous structures in the plant kingdom. Passive traps that exploit geometry, fluid dynamics, and perhaps even the active behavior of their prey to funnel microorganisms into digestive chambers buried in the soil. A typical Genlisea plant is small, often just a few centimeters across, with a rosette of green photosynthetic leaves at the soil surface and slender inflorescences rising above bearing yellow or violet flowers. The flowers are the plant's most visible feature, tubular blooms with a distinctive two-lipped form, the lower lip often larger and more showy than the upper, designed to attract pollinators while keeping the sexual organs out of reach of crawling insects that might otherwise rob nectar without providing pollination services. Below the surface, extending into the wet, peaty substrate, are the rhizophils, translucent, hollow tubes that branch and twist in a characteristic Y-shaped configuration. These structures are ghostly pale, lacking chlorophyll, and they penetrate deep into the soil, sometimes reaching lengths of 10 centimeters or more, threading through the spaces between particles of peat or sand in search of prey. Each rhizophyll has a bulbous vesicle, the digestive chamber, from which extend two arms that spiral in helical corkscrew twists. The arms are lined with narrow slits, the trap openings, arranged along the spiraling length, like windows in a tower. The entire structure is covered with backward, pointing hairs, detentive hairs that angle toward the vesicle, creating a one-way passage. These hairs are crucial to the trap's function. They are flexible enough to bend when pushed from the outside, allowing an organism to enter, but rigid enough to resist when pushed from the inside, preventing escape. The angle at which they project is carefully optimized. Too steep, and they would be ineffective at blocking retreat. Too shallow, and they would impede entry. The result is a surface that feels smooth when touched in one direction, but rough and obstructive when touched in the other. A tactile asymmetry that translates into a directional bias for any creature navigating the trap's interior. An organism that enters through one of the slits can move inward easily, the hairs bending to allow passage, but movement in the opposite direction is blocked. The hairs resist, creating a barrier that becomes more difficult to overcome the harder the organism struggles. It is a lobster pot trap, a passive design that relies not on movement or suction, but on geometry and obstruction to capture and retain prey. The prey of Genlicea are microorganisms, primarily protozoa, such as ciliates and amoebae, though the traps also capture bacteria, algae, nematodes, and small invertebrates. For many years, the mechanism by which these organisms are drawn into the traps was unclear. Early researchers speculated that Genlicea might actively pump water through the rhizophils, creating a flow that would carry prey into the digestive chamber. More recent studies have shown that the plant likely does not generate large-scale fluid flows. Instead, it appears that Genlicea exploits the active swimming behavior of its prey. Bacteria and other microorganisms swim through the soil in search of nutrients. The geometry of the rhizophil, with its spiraling arms and one-way hairs, acts as a bacterial ratchet, a structure that biases the random motion of swimming cells, directing them preferentially toward the digestive vesicle. Once a bacterium or protozoan enters the rhizophil, it is more likely to continue moving inward than to find its way back out. The hairs prevent reversal, and the twisting corkscrew shape of the arms may further confuse the organism's navigation. Gradually, almost inevitably, the prey works its way deeper into the trap until it reaches the vesicle. Inside the vesicle, conditions are hostile to life. The oxygen concentration is negligible, creating an anoxic environment in which most aerobic organisms cannot survive. The plant secretes digestive enzymes, phosphatases that break down cellular structures and release nutrients. The vesicle is also home to a community of bacteria and other microorganisms that contribute to the digestive process. Much as in the pitcher plants, these microbial partners help break down prey tissues and may even serve as prey themselves, their populations continuously replenished by new arrivals through the trap openings. The nutrients released through digestion are absorbed through the walls of the vesicle and transported to the rest of the plant. Because the traps are underground and continuously stimulated by the vast microfauna in the soil, Genlicea secretes digestive enzymes constantly, unlike surface-dwelling carnivores that activate digestion only in response to prey capture. Some species of Genlicea produce two distinct forms of rhizophils, shorter ones and longer ones, which may target different groups of prey or function at different depths in the soil. The genus also exhibits remarkable variation in genome size. Several species, including Genlicea margrete, Genlicea aurea, and Genlicea tuberosa, possess some of the smallest known genomes among all flowering plants, with genome sizes as low as 86 million base pairs, smaller even than some viruses. Other species in the genus have genomes more than 18 times larger. This extraordinary range suggests that the genus has undergone dramatic genomic evolution, with some lineages streamlining their genomes to the bare essentials, while others have expanded through polyploidy or other mechanisms. The reasons for this variation are not fully understood, but it may relate to the specialized ecology of these plants, their unique trapping mechanisms, and the evolutionary pressures they face in nutrient-poor, waterlogged habitats. Across the globe, from the bogs of North America to the wetlands of Southeast Asia, from the mountains of South America to the heathlands of Australia, carnivorous plants occupy a remarkable ecological niche. They are plants that have turned the tables on the animal kingdom. In doing so, they have become integral parts of their ecosystems, playing roles that extend far beyond their own survival. Carnivorous plants are nutrient pumps, concentrating nitrogen, phosphorus, and other elements from prey and making those nutrients available to the broader ecosystem. When the plant dies or when its tissues are consumed by herbivores, they are habitat providers. They're pitchers and traps, creating microhabitats for a diversity of other organisms, from mosquito larvae to bacteria, from spiders to frogs. They are mutualists, forming partnerships with animals that benefit both parties, and they are indicators, sensitive to environmental changes, among the first species to disappear when wetlands are degraded. Their presence or absence telling us much about the health of the ecosystems they inhabit. Yet carnivorous plants are also among the most threatened groups of plants in the world. Approximately 1/4 of all carnivorous plant species, more than 200 species out of roughly 860 known, are at risk of extinction, facing pressures from habitat loss, climate change, pollution, invasive species, and illegal collection. The threats are diverse and often interlinked, creating a web of pressures that few species can withstand. The global diversity hotspots for carnivorous plants are located in regions that are also hotspots for human development: southwestern Western Australia, Southeast Asia, the southeastern United States, Mediterranean Europe, Central Eastern Brazil. In these areas, wetlands are being drained for agriculture, forests are being cleared for timber and development, water sources are being polluted with fertilizers and pesticides, and fire regimes are being altered by both suppression and increased ignition. Many carnivorous plant species are highly specialized, restricted to specific soil types, formed over particular rock formations, adapted to particular elevations, where temperature and moisture conditions fall within narrow ranges, dependent on climatic patterns that have remained stable for thousands of years. When their habitats change, they have nowhere to go, no ability to migrate or adapt quickly enough to survive. The Venus flytrap offers a stark example of these challenges. Despite being one of the most recognizable plants in the world, featured in books, documentaries, and popular culture, the Venus flytrap is native to a remarkably small area, a roughly 100-kilometer radius around Wilmington, North Carolina, in the coastal plain regions of North and South Carolina. It is found nowhere else in the wild. This restricted range makes the species inherently vulnerable. Habitat destruction from residential and commercial development has eliminated many populations. The conversion of wetlands to agricultural land, the building of roads and neighborhoods, the alteration of hydrology through drainage and damming, all have taken their toll. Fire suppression is another significant threat. The longleaf pine savannas and pocosins, where Venus flytraps grow naturally, are fire adapted ecosystems shaped by periodic low-intensity fires that clear away woody vegetation and prevent the encroachment of trees and shrubs. Without fire, these habitats become overgrown, shaded, and unsuitable for Venus flytraps, which require high light levels to thrive. At the same time, fire management in areas near human development must balance ecological needs with safety concerns, making it difficult to maintain the natural fire cycles that the plants depend on. Illegal poaching compounds these problems. Venus flytraps are dug up by the thousands each year from their native habitats and sold illegally. While cultivated Venus flytraps are readily available and inexpensive, there remains a market for wild collected plants, driven partly by the mistaken belief that wild specimens are somehow superior, and partly by the sheer thrill of collecting something rare and forbidden. The removal of mature plants disrupts populations, reducing genetic diversity and eliminating individuals that might otherwise produce seeds to sustain the population. North Carolina has made it illegal to poach Venus flytraps from public and private land, and violations can result in fines and imprisonment, but enforcement is challenging in the remote wetlands where the plants grow. Conservation groups work to raise awareness, educate the public, and support habitat protection efforts. But the future of wild Venus flytrap populations remains precarious. Climate change poses an especially insidious threat to carnivorous plants across the globe. Rising temperatures, shifting precipitation patterns, prolonged droughts, more intense and frequent storms, all impact the delicate hydrological balance of wetlands. Carnivorous plants, which depend on consistent moisture, specific nutrient levels balanced between scarcity and availability, and the presence of stable populations of prey organisms, are vulnerable to even small changes in these conditions. Wetlands are among the ecosystems most sensitive to climate change. They exist at the interface between land and water, maintained by a precise balance of rainfall, groundwater levels, evaporation, and drainage. When that balance shifts, wetlands can dry out, becoming terrestrial habitats unsuitable for species adapted to saturated soils. Or they can become deeper and more permanently flooded, favoring aquatic species over bog and fen specialists. In South Africa, the Cape Floristic region, one of the world's biodiversity hotspots, experienced exceptional droughts from 2015 to 2017. This region is home to 20 carnivorous plant species, including 15 endemics that are found nowhere else on Earth. The drought stressed these populations, reducing flowering and seed production, increasing mortality, and making the plants more vulnerable to other threats. Many of the carnivorous plants in this region grow in small, isolated populations, and the loss of even a few individuals can have disproportionate effects on the viability of the population as a whole. In Australia, the southwestern region harbors more carnivorous plant species than any other place on the planet. A stunning diversity of sundews, pitcher plants, and bladderworts adapted to the region's nutrient-poor soils and Mediterranean climate. But climate models predict that this region will become warmer and drier over the coming decades, with reduced winter rainfall and increased summer temperatures. Many species could lose much or all of their suitable habitat by mid-century, unable to migrate to new areas quickly enough to keep pace with the changing climate. Some species may survive in microrefugia, small pockets of suitable habitat that persist even as the broader landscape changes, but others may simply disappear. The impacts of climate change extend beyond direct effects on temperature and moisture. Changing climate can alter the phenology of plants, the timing of budding, flowering, and seed production, potentially creating mismatches with pollinators or with the seasonal availability of prey. Warmer temperatures can allow invasive species to expand their ranges into areas where they were previously excluded by cold winters. Increased carbon dioxide levels can favor fast-growing, non-carnivorous plants, increasing competition for light and space. Extreme weather events, such as hurricanes, floods, and wildfires, can cause sudden, catastrophic losses of populations. The effects are complex, interconnected, and difficult to predict. But the overall trend is clear. Climate change is making life harder for carnivorous plants. Illegal collection is another serious problem. The unique and fascinating appearance of carnivorous plants makes them desirable to collectors, and some rare species command high prices on the black market. Poaching of wild plants not only removes individuals from vulnerable populations, but also disrupts the ecological relationships that the plants depend on. Venus flytraps are illegally harvested by the thousands each year from their native habitats. Nepenthes species, particularly the rare and spectacular highland species, are collected for sale to hobbyists and enthusiasts. Even well-meaning cultivation can contribute to the problem if it drives demand for wild collected plants. Conservation organizations and governments have worked to regulate trade in carnivorous plants, listing many species under international treaties and national laws. But enforcement is often difficult, especially in remote areas where the plants grow. The complex ecological requirements of carnivorous plants make conservation challenging. Simply protecting the land where they grow is often not enough. Wetlands depend on hydrology, on the flow and chemistry of water, and changes upstream or in the surrounding watershed can degrade a site even if it is legally protected. Fire is essential to many carnivorous plant habitats, clearing away competing vegetation and maintaining the open, sunny conditions that the plants need. Suppressing fires, a common practice in areas near human development, can lead to the decline of fire-adapted species. Conversely, fires that are too frequent or too intense can also be harmful. Restoring degraded habitats and reintroducing extirpated species requires careful attention to soil chemistry, water quality, light levels, and the presence of pollinators and prey. It is delicate work, demanding patience, knowledge, and resources. Despite these challenges, there are reasons for hope. Carnivorous plants have captured the public imagination in ways that few other plant groups have. They are charismatic, accessible, easily understood even by children. This popularity has translated into support for conservation efforts. Botanical gardens around the world maintain collections of carnivorous plants, preserving genetic diversity and providing stock for reintroduction programs. Researchers are studying the ecology, genetics, and physiology of these plants, uncovering the secrets of their adaptations and informing conservation strategies. Community science projects engage volunteers in monitoring populations and reporting sightings. Legal protections are being strengthened, and awareness is growing about the importance of wetlands and the threats they face. Some populations of once rare species are recovering, Restoration projects are bringing carnivorous plants back to areas where they had disappeared, and there is something profoundly inspiring about these plants. Something that speaks to the resilience and creativity of life. Carnivorous plants are survivors, inhabitants of the most marginal lands, places where the soil offers almost nothing and competition from other plants is fierce. In response, they have evolved strategies that are as elegant as they are deadly, turning the very challenges of their environment into opportunities. They have become predators, hunters, creatures that defy our expectations of what a plant can be. In the stillness of a bog, in the slow passage of seasons, they wait, patient and watchful, their traps set, their enzymes ready. When prey arrives, they respond with movements that rival the fastest animals, with mechanisms that exploit physics and chemistry in ways that continue to amaze scientists. They remind us that evolution is endlessly inventive, that life finds a way to flourish even in the harshest conditions. Adaptation is not just about survival, but about transformation. As you drift deeper into sleep tonight, you might imagine yourself standing at the edge of a bog at dawn. The air cool and still, mist rising from the water, the light soft and golden. Around you are the carnivorous plants, their pitchers glistening with dew, their sundew tentacles catching the first rays of sun, their Venus flytraps open and waiting. Beneath your feet, hidden in the wet soil, Bladderworts and Corkscrew plants hunt in silence. Above you, the Waterwheel plant drifts in a shallow pool, its delicate traps poised to snap. This is a world both beautiful and strange, a place where plants have become predators, where the usual rules are upturned, where patience and adaptation have created wonders beyond counting. And in this place, there is peace. The bog is alive with subtle movements, with quiet transformations, with the slow turning of seasons and the steady work of evolution. It is a reminder that the world is full of mystery, that there are still things to learn, connections to discover, relationships to marvel at. And as you stand there, breathing in the cool morning air, you feel a deep sense of calm, a recognition that you are part of something larger, something ancient and enduring. The carnivorous plants, with their intricate traps and their quiet persistence, have lessons to offer about adaptation and resilience, about finding nourishment even in scarcity, about thriving in places where others cannot. These are lessons worth carrying with you as you rest, as you let go of the day's tensions, as you allow yourself to sink into the comfort of sleep. If you found wonder and relaxation in this journey through the world of carnivorous plants, please consider subscribing to the channel and leaving a like. It helps others discover these stories, these moments of peace and learning. And as you close your eyes and let sleep come, know that the world of plants continues its quiet work, evolving, adapting, surviving, a testament to the enduring creativity of life on Earth. Rest well, and let the gentle knowledge of nature's ingenuity carry you into your dreams.